Although it is extremely difficult to pinpoint the precise beginning of human awareness of the presence and role of microorganisms in foods, the available evidence indicates that this knowledge preceded the establishment of bacteriology or microbiology as a science. The era prior to the establishment of bacteriology as a science may be designated the pre-scientific era. This era may be further divided into what has been called the food gathering period and the food producing period. The former covers the time from human origin over 1 million years ago up to 8,000 years ago. During this period, humans were presumably carnivorous, with plant foods coming into their diet later in this period. It is also during this period that foods were first cooked. The food-producing period dates from about 8,000 to 10,000 years ago and of course includes the present time. It is presumed that the problems of spoilage and food poisoning were encountered early in this period. With the advent of prepared foods, the problems of disease transmission by foods and of faster spoilage caused by improper storage made their appearance. Spoilage of prepared foods apparently dates from about 6000 BC. The practice of making pottery was brought to Western Europe about 5000 BC from the Near East. The first boiler pots are thought to have originated in the Near East about 8,000 years ago. The arts of cereal cookery, brewing, and food storage were either started at about this time or stimulated by this new development. The first evidence of beer manufacture has been traced to ancient Babylonia as far back as 7000 BC. The Sumerians of about 3000 BC are believed to have been the first great livestock breeders and dairymen and were among the first to make butter. Salted meats, fish, fat, dried skins, wheat, and barley are also known to have been associated with this culture. Milk, butter, and cheese were used by the Egyptians as early as 3000 BC. Between 3000 BC and 1200 BC, the Jews used salt from the Dead Sea in the preservation of various foods. The Chinese and Greeks used salted fish in their diet, and the Greeks are credited with passing this practice on to the Romans, whose diet included pickled meats. Mummification and preservation of foods were related technologies that seem to have influenced each other's development. Wines are known to have been prepared by the Assyrians by 3500 BC. Fermented sausages were prepared and consumed by the ancient Babylonians and the people of ancient China as far back as 1500 BC. Another method of food preservation that apparently arose during this time was the use of oils such as olive and sesame. Jensen has pointed out that the use of oils leads to high incidences of staphylococcal food poisoning. The Romans excelled in the preservation of meats other than beef by around 1000 BC and are known to have used snow to pack prawns and other perishables according to Seneca. The practice of smoking meats as a form of preservation is presumed to have emerged sometime during this period, as did the making of cheese and wines. It is doubtful whether people at this time understood the nature of these newly found preservation techniques. It is also doubtful whether the role of foods in the transmission of disease or the danger of eating meat from infected animals was recognized.
Few advances were apparently made toward understanding the nature of food poisoning and food spoilage between the time of the birth of Christ and AD 1100. Ergot poisoning caused by Claviceps purpurea, a fungus that grows on rye and other grains, caused many deaths during the Middle Ages. Over 40,000 deaths due to ergot poisoning were recorded in France alone in AD 943, but it was not known that the toxin of this disease was produced by a fungus. Meat butchers are mentioned for the first time in 1156, and by 1248, the Swiss were concerned with marketable and non-marketable meats. In 1276, a compulsory slaughter and inspection order was issued for public abattoirs in Augsburg. Although people were aware of quality attributes in meats by the 13th century, it is doubtful whether there was any knowledge of the causal relationship between meat quality and microorganisms. Perhaps the first person to suggest the role of microorganisms in spoiling foods was A. Kircher, a monk who as early as 1658 examined decaying bodies, meat, milk, and other substances and saw what he referred to as worms invisible to the naked eye. Kircher's descriptions lacked precision, however, and his observations did not receive wide acceptance. In 1765, L. Spallanzani showed that beef broth that had been boiled for an hour and sealed remained sterile and did not spoil. Spallanzani performed this experiment to disprove the doctrine of the spontaneous generation of life. However, he did not convince the proponents of the theory because they believed that this treatment excluded oxygen, which they felt was vital to spontaneous generation. In 1837, Schwann showed that heated infusions remained sterile in the presence of air, which he supplied by passing it through heated coils into the infusion. Although both of these men demonstrated the idea of the heat preservation of foods, neither took advantage of his findings with respect to application. The same way may be said of D. Papin and G. Leibniz, who hinted at the heat preservation of foods at the turn of the 18th century. The history of thermal canning necessitates a brief biography of Nicolas Appert. This Frenchman worked in his father's wine cellar early on, and he and two brothers established a brewery in 1778. In 1784, he opened a confectioner's store in Paris that was later transformed into a wholesale business. His discovery of a food preservation process occurred between 1789 and 1793. He established a cannery in 1802 and exported his products to other countries. The French Navy began testing his preservation method in 1802, and in 1809, a French ministry official encouraged him to promote his invention. In 1810, he published his method and was awarded the sum of 12,000 francs. This, of course, was the beginning of canning as it is known and practiced today. This event occurred some 50 years before L. Pasteur demonstrated the role of microorganisms in the spoilage of French wines, a development that gave rise to the rediscovery of bacteria. A. Leeuwenhoek in the Netherlands had examined bacteria through a microscope and described them in 1683, but it is unlikely that Appert was aware of this development, and Leeuwenhoek's report was not available in French. The first person to appreciate and understand the presence and role of microorganisms in food was Pasteur. In 1837, he showed that the souring of milk was caused by microorganisms, and in about 1860, he used heat for the first time to destroy undesirable organisms in wine and beer. This process is now known as pasteurization.